So now I'm very happy to present the last keynote speaker of the entire conference. So Dr. Gisela Hagberg did a master's in engineering physics at the EPFL Lausanne in Switzerland and at Lund University in Sweden. She pursued a PhD in biophysics at the University of Basel, Switzerland, and in medical physics at the Sapienza University in Rome, Italy. Currently, she works at the biomedical magnetic resonance at the University of Tübingen in Germany, and is also affiliated with the high-field magnetic resonance department of the Max Planck Institute for Biological Cybernetics in Tübingen. Her research interests lie in the field of neuroimaging applications, from MR physics and image reconstruction to data analysis and limits of MRI interpretation. Today, she's going to present on the topic of quantitative susceptibility mapping, where she will highlight potential pitfalls and clinical applications. So please, Dr. Hagberg, we are very eager to hear you.
So maybe you can hear me now. Can you hear me?
Yes.
Yes. Can you see my screen?
Not yet.
Not yet. Okay. So, okay. Now it should be working.
Yes, it's good. Thank you.
Good. Thank you so much to all the organizers for this beautiful congress and for inviting me. And thanks also to you, Evgenia, for this beautiful talk. This talk will also be concentrated on iron, I would say, because when we ask the question, how quantitative is quantitative susceptibility mapping, at the time being, at the standard of the research right now, we basically concentrate our efforts on gray matter nuclei, gray matter, also brain regions which are dominated by gray matter, although some groups are also working with white matter QSM, and that is much more challenging, I'd say, and I will not go into that. What I will concentrate on is how can we validate this technique? Because fact is that quantitative mapping with MRI is quite amazing. It's a fundamental parameter for MRI, and it shows no field dependence in contrast to what R1 or R2 does. It can separate between dia and paramagnetic effects, and that R2 cannot do for us. So it has been used in the literature to characterize calcifications, to determine increase of iron in neurodegeneration, to look at oxygenation, but it has also been used to segment veins or Alzheimer's plaques. And this technique has been around 15 years, basically, and a large community is using it and is driving the evolution of this technique forwards, but still it's not really a clinical valid or ubiquitous method as of yet. And my talk is trying to walk you through some of these factors that may be causing that, and maybe point towards ways that we can use different tools to validate the protocols and the post-processing that we are working on at different groups to help to overcome that limit. So I will talk to you a little bit about magnetic susceptibility, about measuring QSM using MRI, and then different tools for validating it in vivo, ex vivo, using open resources and using QSM phantoms. And a bit of a conflict of interest, I guess. My main interest lies in the high field, high resolution MRI. Okay, magnetic susceptibility. If we would just have a magnetic field in vacuum, there will be no magnetic induction, so to say. We will only have the direct magnetic field that we apply. But as soon as we put some matter in here, things will change. So the matter itself will change the local magnetic field dependent on whether it's diamagnetic or para or ferromagnetic. And we can describe this using magnetostatics, so using Maxwell's equations, which then relates the external field and the electric field of the matter. So the induced field that eventually will be the object that we actually measure with our MRI scanners, it depends on the magnetic susceptibility of that matter. And that is also what gives us our magnetization. So there are different types of magnetic susceptibilities, and we can separate the two most likely ones to be found in vivo, and that's diamagnetic and paramagnetic species. So diamagnetism does not show any temperature dependence, and it purely depends on the electron cloud which surrounds the nucleus and its distance from the center of the nucleus. And it will be like an effect like Lenz's law. So basically these electrons, they try to protect, let's say, the nucleus from the external field and make the field smaller at the level of our hydrogen spins that we are looking at. Paramagnetism, on the other hand, it shows temperature dependence. And so its effects then increase at lower temperature. And that is a characteristic which can be used with other techniques than MRI to pinpoint its magnetism. Another way of looking at paramagnetism is to vary the field and see how much field is inside the object. And this you can, for instance, do with vibrating sample magnetometry. And if you do that for some iron oxides, as we have done here, you will see these beautiful Pango-superporicus curves. And these are particularly beautiful for well-behaved paramagnetic species like the lanthanides. So gadolinium is a lanthanide, a rare earth metal, and it behaves perfectly, and you can predict perfectly its magnetic moment. This is not so true for iron and iron oxides in their different forms that we can find in vivo. But generally speaking, there will be some kind of saturation magnetization at high fields. They are typically, we have 3-4 Tesla. So if you work at 3 Tesla and above, your iron is likely to be saturated. So all biometals that we have in our body, they have different magnetic susceptibility. And if you go to one of these beautiful resources and look at what values we can find, these values are usually valid for solid state situations. And they do also not consider what happens when these atoms come into play in a molecule, or if these molecules are ordered in different structures. And all these ordering effects will also influence what we actually can see with an MRI scanner. And this was noted already in 1992, actually, in Schenk's beautiful paper about magnetic susceptibility, which I warmly can recommend. So there are several relevant factors for susceptibility at the atomer and the molecular level. So we have seen the spin state, but also the oxidation state, the carrier molecule, and the particle size order, basically everything which somehow influences how the electrons are distributed around our magnetic resonance sensitive nuclei. So let's now take a step into the scanner. And here I have listed something which is very, very familiar to most of you, assuming that you are MRI people. So we have on your left hand side the MRI scanning acquisition protocol, and your right hand side the post-processing. And the factors that I have listed here, which basically takes everything into account that we actually use when we do MRI, can influence the values that we get with QSM. So attention is on the water when we optimize these different steps. So optimizing a protocol and bringing it to useful application may entail keeping track of and observing closely all these different factors. And what you see on your right-hand side, I will not talk so much about today, a little bit, but actually there is a quite fantastic literature about these different steps. So how the scanner has its own sign of the face. It can be clockwise or anti-clockwise. How you should combine the face maps from different coils in the best way to optimize really the face signal. How using brain tissue masking, I will show you some examples of that. How you can unwrap that face because it will be aliased into the 0 to 2 pi range. How you can do background correction and then eventually arrive at your QSM images. So if you're fortunately, you will have a background corrected field map. And if you have that, then you can use the dipole approximation, which lies at the very heart of the QSM method. This means that in image space, you would deconvolve your image using this dipole that you can see on the right. Unfortunately, there is a more handy way of doing it by multiplication, and that is done in K-space. And when that was understood by the scientific community, QSM took off, and all these beautiful methods that we have today that are also available in programs and toolboxes that are readily available came out. So there is one caveat of this method, and you can see that on the location in K-space at the so-called cone of zero coefficient. Basically, the dipole is not defined there. So it's since you cannot measure there without actually rotating your object, you cannot make a good estimate from just one single orientation measurement without doing a lot of regularization during calculation. And that can also be one matter of taking care of when you do QSM. The second thing which lies at the very heart of QSM is this concept of sphere of Lorentz, and that has been beautifully reviewed by Durant in Concepts of Magnetic Resonance in 2003. And here I have taken from Schweser's paper a cartoon which demonstrates how the field, the total demagnetizing field, because the object that you put in your field actually changes the field not only inside itself but also outside. So you have to consider that on the border between the object that you would like to study and its outside, the relation between the magnetic field and the internal must be in homeostasis, so to say, or in equilibrium. And therefore, you must take this concept into consideration, and it is basically inside the algorithms that you use for the calculation. So if this concept is no longer valid, in principle, your QSM would not be quantitative anymore. So just to put it in more straightforward terms, when is it valid? Well, we assume that one voxel is one single, very homogeneous susceptibility source, which is not true, because as you can see here on your right-hand side, it shows how iron is compartmentalized inside a neuron. We saw before in Kirillina's talk how iron can be compartmentalized in other ways in the tissue. So this is basically an assumption that we would need to make, but which is not reflected in tissue in vivo today. The other thing we just assume is that susceptibility is isotropic. And this is not true. This was recognized very early here by Chun-Lei Liu, who showed that susceptibility, especially in white matter fibers, is anisotropic and shows orientation dependence. A third point is that we actually would need to measure MRI at multiple orientations, so at least 3 orientations to cover that zero cone. But usually we only have one orientation. Another thing which we kind of skipped before is we need background corrected images. That's also a tricky bit, because a part of the information that we would need we have no information from, and that is the outside of the head. So in order to really measure the background field, we would, I guess, need to put a snorkel in the mouth of our volunteers and put them in a water bath, and we would have protons outside the head. But we don't have, so we really need to define a red area here, a region of interest, for which we can perfectly correct the background, and then we can get our susceptibility distribution. And this red region of interest you see here is of utmost importance. It's our tissue mask or tissue brain mask. And there are different ways we can do that. Here is some example, and which shows the interplay between the background correction and tissue masking. So on your very left-hand side, you see the resharp filtering in a very, very strongly regularized version. And you see that dependent on whether, or regardless of the masking, the images look pretty good. If you move towards the right-hand side, we have more soft background corrections, and there you see huge effects if you do not do the masking in a good way. Here we have used phase-based information to complement the magnitude-only signal as described in our paper last year. So it may have a role. So now, let's come to the juicy bits. Validation. How shall we validate our QSM measurements? And how can we do that at each single site? One possibility is to use, well, the human brain. And that has great advantages. We can test different acquisition protocols. We can look at the background field. We can also have tissue susceptibility all there in one, and it's a real realistic situation. The back downside, as you soon will see, is the question whether or not we really have a gold standard. One possibility is to use our favorite paper. It's this paper from 1958, where Hagberg and Surander showed how the iron content increases over age. And so basically you can either gather persons with different ages, or take the same persons and look at how the QSM signal looks in different brain regions, because the iron content shows differences there, and it's all perfectly modeled by this beautiful exponential functions here. So it's very handy for prediction and adaptation to the routine life in doing QSM. Another thing is, however, our gold standard. So ferritin is assumed to account for most of the iron in our brain. So what about ferritin then? Well, it's loaded with, well, it was cited 4,500 iron atoms, and it has a magnetic moment of 3.78. As you see on your right-hand side, Brooks et al. Actually tried to measure this, and what they found is that due to the packing of the iron inside the ferritin molecule, or the ferritin container, that magnetic moment will actually go down to 2.9. And maybe a bit more cumbersome than this is that this 4,500 atoms is really not the maximum value. And it comes from a miscitation in the scientific field. And I can warmly recommend this paper by Hagen and Metalonics, which shows how citation bias can bring us to actually assume wrong things about what we are actually measuring. So sometimes it can be good to look deeper into literature. So a more likely value for the maximum ferritin loading is 4,300 atoms. And the question is now, can we see it in vivo with QSM? So here's an example of that. So here I've taken data from the paper from Andreas Dijston from 2013 and taken the magnetic susceptibility values in different brain regions. And I knew the age of this volunteer, so I could, from Hagberg and Solander's models, tell how much iron there is likely to be in there. And you see, you can find values there, and I come to 36 parts per billion per millimolar iron. So if I look into the literature, there are a wide range of different values in vivo, ex vivo. So maybe it's hard to use the in vivo QSM contrast as a gold standard. But okay, let's try it and see what happens with our tissue masks. So here is an example. You can see here in the middle how ReSharp or WooSharp or LBV background filtering can actually change our QSM contrast quite a lot. So if you use the strong filters, which don't care about the brain tissue masking, we actually get a too low QSM contrast. But if we fix the masks a little bit, we can come more close. We don't need such strong filters, and we can come more closest to the more likely gold standard. Basically, the take-home message here is that also the background field strengths correction can influence the actual contrast that we measure. On the right-hand side, I would like to point to the facts that we can actually avoid echo time dependence also by checking the masks and work more with that. How about ex vivo? Can we use that as a validation? Well, perhaps yes. So in this paper, where we studied the effect of formalin fixation on ex vivo specimens over time, actually QSM was one of the more stable ones. After 5 days, we have a strong paramagnetic shift, but then it stabilizes and it seems quite okayish after a while. Now, a third way of doing validation is to use open resources. And the QSM people are very, very lucky to have an active community who organizes challenges and organizes data that you can download and actually use and check how your methods perform in comparison with other groups' methods. And this is a beauty. So here's an example of that, where the 2016 challenge data was used, and there has been a challenge number 2, which then used an in silico model for QSM in the brain. And that is really, really helpful. There is one caveat, though. We are not actually measuring the data ourselves. And if we also want to check our scanner and their protocols, this is maybe not an option. And then we need to look further. One possibility is to use contrast agents. So gadolinium is a beautiful susceptibility source, and here's an example of that. And it has been used both in multi-center studies and in single-center studies. And we can actually introduce head-shaped containers to obtain the background field, which has been missing in paper so far. So we already saw this, relevant factors at the atomer and molecular level for the susceptibility. How about MRI? Well, there, the macronesoscopic scale comes into play. Not only the concentration makes a difference, but also the spatial distribution of our iron, and also diffusion of water through field gradients. So maybe there is a call for more complex phantoms, and this is emerging also in the QSM field. And I'd like to present one of these phantoms to you today, and it's basically this one. It's iron-loaded sodium polyacrylate clusters that we have packed in alginate to make it more stable. And you can see a photo here on the right, and it basically leads to this snowflake-like non-uniform distribution of susceptibility. And we have put some effort into validating it and checking if it's actually useful in multi-center studies. And we compared 2 forms of iron-free and clustered iron and validated it with vibrating sample magnetometry and found that it performed quite well at 4 different machines. And we also have applied it used it at 14 Tesla, and it actually seems to work. Here are some examples of that. So it can be handy to study voxel size effects for QSM. To try to more respect the underlying assumptions of QSM by moving toward smaller voxel sizes. And it can be used to validate some clinical application and maybe answer the question if it's worth doing QSM when our voxel sizes are so big in vivo. And one example of that is the study of something very small. So let's take something very small. These are corpora amylacea. They're basically some polyglucan proteinations, little beads sitting in the brains, and you can see them ex vivo in elderly subjects. And they're quite small, 10 micron, 20 micron. And we can see it beautifully in microscopy by using different types of stains. Here I'm showing periodic acid shift stains, which are specific for this type of objects. But you can also see a myelin stain here, actually, because, yes, they are diamagnetic. So we needed to check if there is not some funny myelin accumulation going on here. They can shine up as bright little spots in R2-star, and in QSM they are diamagnetic. And here we measured R2-star and QSM with voxel sizes of 200 micron isotropic, and so it's a factor of 10 bigger than these actual small corpora amylacea, but they tend to accumulate, so we can still see them. Another example of this is voxel size effects in Alzheimer's disease. So here is a small simulation from a beta amyloid stain that then had been forward modeled to see the magnetic field at different voxel sizes. And Elisa Tutsi, she came up with this method that we can actually use this technique and try to estimate the amyloid beta load ex vivo. So now we can then go back and we can take our non-uniform iron phantoms and see if that technique also works there. And here are some examples. You can see images with different voxel sizes. You can see the histograms for different voxel sizes. And here is the volume fraction that we determined. So for very low iron concentration, it actually seems to work quite nicely. So in conclusions now, QSM, it has many pitfalls. MRI acquisition and post-processing can make a difference at the end of the day, and it may be worth a while to invest some time and look into the nitty-gritties. We have several validation tools at our hands. We can use in vivo, likely also ex vivo MRI. There are these beautiful challenge resources that we can use, and there are QSM phantoms coming and available that can be helpful. And in future studies, I expect to see more about local non-uniform distributions of bimetals in the tissue, possibilities to look at source separation, as we saw in last year's QMRI talk by John Lee, and also studies looking at more complicated forms of iron and diamagnetic sources in the myelinated axons. And with that, I would like to thank you all and thank our fundings and all the co-workers, students and collaborators here in Tübingen and abroad.
Thank you so much. It was really interesting. Mark, do we have any questions in the chat?
Yes, we do. So from Olivier Perrant, he asks, how close are we to being able to reliably estimate QSM in the cortex?
Yes, so thank you. That's a very good question. So a little bit with the issue with the cortex is that the mask that many use eats up a lot of the cortex, because the cortex is very, very close to the cranium, and there are strong susceptibility differences in that area. You can be cautious and have a better look at it, and we found that these phase-based masks were quite helpful to help looking at it, and it actually seems to me that it works pretty well.
Okay, all right, thank you. And then from Dorian Pustina, they say, thank you for the excellent talk. What are the advised steps required to include QSM in multi-center trials? Do we still need to balance patient groups between sites after harmonizing QSM acquisitions?
That's a very, very good question. So the ideal thing for me would be that it would just suffice to use the phantom, and then you would be comparable. Now, this has been said also for T1, for instance, and T1 has then, in the end, when people, the T1 and T1 weighted images is the thing that are mostly widely used in multi-center studies, and there are huge problems, and there are incredible differences between sites, also for something as, let's say, simplistic as T1 weighted imaging. So I wouldn't be surprised if that would also be the case with QSM. In principle, it should work, but I wouldn't put my hand on it. I would be thrilled to see.
Okay. All right. Thank you. Very interesting. There's a few more here, but perhaps we should move on and save them for the discussion.
Amazing. Thank you again for your great talk.